Thank you as well for Azure for sponsoring this session, and I want to thank as well all the audience for attending this meeting. Today I'm going to be talking about the specialized lipid mediators, drivers of inflammation. This is my disclosure slide, or what I also call the thank you slide, because without these centers, companies, and products, none of this will be possible. This is the outline of my talk for today. And today, many diseases are related in part by inflammation, and especially with uncontrolled inflammation. Here are listed some of the major diseases, and we can see in the center that all of them converge to a pro-inflammatory component. So the new villains nowadays, we could consider these two, the inflammation and the pro-inflammatory diet. Professor Serhan asks what turns inflammation on. And studying self-limited acute inflammatory response models learned that resolution of inflammation is actually an active process. And it is this family of lipid mediators that brings us to this state of resolution and homeostasis. These chronic diseases are caused in part by a variety of poor or excessive habits such as stress, inadequate physical exercise, alcohol, tobacco, or poor nutrition. What is common on all these diseases on the right is a deficiency in a specialized pro-resolving mediator, or SPMs, here in the center. I want to point as an example the work from John Claria's lab, where they concluded that by addition of monohydroxylated 17-DHA intermediate mediator, increased synthesis of resolvins by pro-inflammatory leukocytes from obese patients. This slide shows the decision path for an acute inflammation and can be chronic or resolution. And obviously, the ideal outcome is resolution. So after an injury or an infection, there is a synthesis of different chemical mediators that activate acute inflammatory response that may or may not follow with the cardinal signs of inflammation, such as calor, rubor, tumor, dolor, and loss of function. And historically, the termination of inflammation was thought to be passive, a passive process. That by diffusion of dilution or these chemical mediators and eventually finish the inflammatory process. But Professor Serhan discovered this superfamily of lipid mediators called specialized pro-resolving mediators that are actively synthesized to finish inflammation. If this is disrupted, can chronify inflammation, and then diseases can appear. So over time, we learn from textbooks that after an inflammatory response, there is an initiation phase, which is protective for the host, and then starts the resolution phase, which is SPMs are key players in bringing in inflammation down. If this process is disrupted, then inflammation can be chronified, and there is a loss of resolution and function. SPMs act directly limiting neutrophil infiltration to the rolling process and diaperesis, blocking inflammatory cytokines. And eicosanoids, as you can see here on the bottom right. As a consequence, limit systemic effects like fever and pain, etc. This graph shows a typical self-limited acute inflammatory response time course. From the left, after an injury or infection, there is an edema formation followed by a rapid infiltration of neutrophils that will become apoptotic, and then infiltrating monocytes and macrophages, shown here in blue, will clear these apoptotic neutrophils, leading to resolution. Using self-limiting inflammatory models, Professor Serhan demonstrated a temporal biosynthesis of SPMs in the resolution phase. These SPMs are a family of endogenous mediators, including lipoxins, resolvins, protectins, and merisins. These mediators promote the signs of resolution listed here in blue. Identification of these SPMs provided the first evidence that resolution of inflammation is an active process. SPMs are derived from EPA, DHA, and DHA, and are classified by different families, the resolvins, the protectins, the merisins family. and they are biosynthesized in the organisms through a complex multi-step conversion pathway that involve different enzymes, cyclooxygenases and lipoxygenases, etc. So how do SPMs work? What is the mechanism of action? This illustration shows a post-capillary venule and illustrates how SPMs are limiting the magnitude and duration of the acute inflammatory response by limiting the further recruitment of neutrophils, but stimulating the non-logistic recruitment of monocytes. And then when macrophages encounter the SPMs, increase phagocytosis and efferocytosis to remove debris and apoptotic cells, as well as killing microbes. It's using these resolution indices in experimental setting introduced by Serhan that gives the mechanisms of action of a specialized pro-resolving mediators and allow us to quantify and study how they accelerate resolution. These SPMs are known to counteract and regulate the early initiatives of acute inflammation and the prostaglandins. They regulate COX-2 expression, leukotriene synthesis, chemokines, and cytokines. They also upregulate anti-inflammatory molecules like IL-10 and ultimately regulate edema as well. The SPMs has been proven in multiple in vivo experimental models that are not immune suppressive, and these are key actions for these mediators. This slide shows the relative potency of different mediators, and you can see on the bottom left in blue that SPMs are in the range of nano to picomolar potency. By comparison, in this range, that is where neurotransmitters carry their actions. So these mediators are extremely potent and are agonists of resolution. There has been identified 6 GPCR receptors, and there are these stereoselective receptors expressed in different cell types that triggers its intracellular signaling pathways that ultimately will trigger the pro-resolving functions. Most recently, we identified LGR6 to be a merism-1 receptor published in this journal, JCI, by Dr. Nan Chang in 2019. So today, several labs in the world, more and more, are incorporating these lipid mediators targeted metabolipidomics that allow us to profile tissues and fluids in order to determine the lipid mediator profile and quantify its lipidic content. For example, we are building a database where we can store different profiles of organs and in order to study for future clinical trials. So using this approach, we have learned now that many human tissues have been shown to produce pro-resolving mediators. And I will emphasize this breast milk work from Dr. Hildur Arnardóttir, when she was a postdoc in Serhan's lab, as well as the high levels in placenta from the guys from Australia, or the work in adipose tissue and obesity from John Sclaria's lab in Barcelona. I want to finish with these slides, and I hope I convince you that this superfamily of lipid mediators are the circuits that drive resolution of inflammation, that SPMs evoke the signs of resolution and they counteract the cardinal signs of inflammation. So my summary slides. Active resolution of inflammation is a process governed by SPM, stimulating signs of resolution. SPM actions and mechanisms are potent and stereoselective bioactions and not immune suppressive. And SPM receptor axis stimulate immune functions, protects organs, and control infection. Altogether, SPMs, their pathway and receptors provide new opportunities to control unwanted inflammation, infection, and reduce organ injury, enabling the potential of resolution physiology and pharmacology. I want to finish. Thank you to all the labs members from Serhan Lab, as well as Professor Serhan, Jeremy Winkler, Nan Chang, Paul Norris, Stephanie Oliveros, and Charlotte Joubin. Thank you very much.